Okay. Hi everyone. This is not working.
Why say hi everybody? Why say
There we
hi
go.
to everybody?
Oh, sorry guys. We have an extra participant here. Say hi. Okay, sorry about that. My YouTube was doing very weird things. I'm going to turn on extra light here. This is not proving to be as bright as I thought. So sorry. Give me 2 seconds. Baby, you want to say hi to them? Hi. Okay, sit up for a second, okay?
Where's them? That's better.
That's better. Okay. Okay. All right. So I'm so sorry. We had technical difficulties and a non-sleeper. But go ahead and fire away any questions you have, and I'll go ahead and answer them. Maybe she'll fall asleep as we're doing this because she doesn't want to sleep. Okay, so what questions do you guys have about cellular respiration? Anything?
I see your face on the computer.
I know. You can see yourself too. See, there you are. Doesn't look like we have a huge crowd right now. That's okay. Can you guys? Okay. Well, I guess I'll just start talking and see what happens. Baby, go sleep. Okay. For cellular respiration, photosynthesis, one thing people have asked is like, do they need to know the exact numbers of things, like ATPs and whatnot? You do not. I want you to know the basic idea of what happens. That's our house, baby. So you need to know like the idea of what happens in each process. I'm not at any given time going to tell you or ask you to like tell me exactly what happens, like in terms of numbers of ATP and whatnot, but you should definitely know like which processes yield more ATP and things like that. Okay, it's okay, you sleep. Okay, good. But like I said, what questions do you guys have? Anything? Because I'm not really sure what we should talk about. But I can see it if you type something. Actually, can someone just test something out and type something just to make sure I can see it? Any of you who are watching? Oh, hi, Audrey.
We say hi, Auntie.
I'm saying hi, Audrey, because she helped me type something to see if I can see anything. Okay. Let me see, what else can I talk to you guys about? Make sure you know each of the processes of cellular respiration and photosynthesis. Like I said, I want you to know reactants. I want you to know products. You should understand the processes by which they happen. But you don't need to know the individual. You don't need to know the individual like enzymes and things, right? Like I'm not going to ask you to reproduce the entire process.
I did it. One more piece. One more piece for the computer.
Oh, okay. Thank you.
We lost it.
No, I didn't lose it. Thank you. Let's see. Know the difference between aerobic and anaerobic processes. Oh, okay. Sure. Thank you, Audrey. She's asking me questions, so mommy doesn't have to keep talking to myself. Yes, so alternate forms of photosynthesis. The idea behind it is that every plant has rubisco, right? Every plant is going to use the Calvin cycle to generate the intermediate G3P that's going to ultimately make glucose. But the alternate forms are the ones who now add an extra step in front of that process to make it more feasible. Yes, you see your hand. Okay. So for example, rubisco, like I said, does both reactions, oxygenase and carboxylase, right? But we want to optimize carboxylase reactions happening and not oxygen reactions. No, thank you, baby. You need to go sleep. Can you rest, please? So with C4 photosynthesis, which is two different types, there's C4 special anatomy, and there's C4 special anatomy, and there's the CAM pathway. Those are both going to be processes in which they add an extra step in front of the Calvin cycle so that they can, thank you. So, okay, thank you. Shh. So they can have a better chance of trapping the CO2 and not doing the oxygenase reaction. So with the C4 special anatomy pathway, that's where they have the bundle sheath cells, right? Where they essentially do the light reactions in the mesophyll cells and then use PEP carboxylase to trap the carbon dioxide into a 4-carbon compound. After they do that, then they can essentially smuggle that 4-carbon compound into the bundle sheath cells and basically create I shouldn't say artificial, but create a situation in which the rubisco in the bundle sheath cells really only has mostly carbon dioxide, not very much oxygen. She's sort of forcing it to react with carbon dioxide. Whereas the CAM pathway is also an alternate form, also C4 photosynthesis, also converting carbon dioxide into a 4-carbon compound. But that difference is it's the Costco method, right? You do it at nighttime. So in doing so, it's not going to be able to, it will basically be able to close its stomata during the daytime. So it doesn't really have to worry about running out of CO2 in the daytime, even though it's closing its stomata. So that's your C4 photosynthesis, your alternate pathways. But again, remember though, rubisco is still there. It's just like adding an appetizer in front of it. So yes, Sonia, like I said, the C4 and CAM pathways are both, thank you, are both C4 photosynthesis. They are both processes in which we have a 4-carbon compound being the way that you're trapping carbon dioxide instead of rubisco. So in doing so, because PEP carboxylase is so much more attracted to carbon dioxide, it doesn't really want to react with oxygen. You're going to have it pretty much much more likely that the carboxylase reaction happens instead of the oxygenase reaction. The only difference between the C4 and CAM pathways is sort of how they do that, right? The C4 pathway will do it as a matter of separation of space, trap the CO2 in one cell, but then shuttle it into another one to actually do the Calvin cycle, whereas the CAM pathway has the same anatomy as a C4 plant, or sorry, C3 plant, like a regular photosynthetic plant, but instead it's going to trap it at night and then use it during the day. Yes, I can totally go for light and dark reactions. So remember, the light reactions are the first part of photosynthesis. In those reactions, it's the idea that that's where you're really getting the energy or converting the sunlight's energy to allow you to, or not you, because we don't do this, to allow plants to do photosynthesis.
You see my ear.
Yes. So with the light reactions, that is where essentially first, this is in the thylakoid discs in the chloroplast, and they are going to, the light shines down on the chloroplast. This is the reaction. These are the things we did in class with the board demo, where now the photons of light strike the chlorophyll, photosystems 1 and 2. And when we strike photosystems 1 and 2, then the electrons in there get excited. They jump to a higher energy level. In doing so, the number goes photosystem 2, then photosystem 1. So the electron from photosystem 2 goes down an electron transport chain, and that electron transport chain produces ATP. The second photosystem, which is actually photosystem 1, its electron gets excited, goes down to a smaller electron transport chain. That's going to make NADPH. With the dark reactions, remember that's called the Calvin cycle, and that is where now you are going to use the ATP and the NADPH that was created in the light reaction. So it's not that you actually need darkness, you just don't need light directly, right? But now the idea is that the Calvin cycle, the rubisco is going to trap the CO2, take it through a series of reactions, and in doing so, Okay. In doing so, you are going to end up with um, the essentially with you first trap and this will actually address your question, Somia. Rubisco is the enzyme. RUBP is the 5 carbon molecule. So each CO2 gets trapped, which gets incorporated into a 6 carbon molecule, which then splits into 2 3 carbon molecules. So RUBP is the 5 carbon molecule that you join CO2 to. So essentially, Rubisco is the enzyme, and RUBP is what Rubisco attaches CO2 to. Yes, Audrey, I can go over the. No, you don't need to know intermediates. I mean, I would want you to know Rubisco. RUBP, I would say, is maybe the minimum intermediate you should know, but like all the stuff in between, I don't need you to know. Okay, so feedback mechanisms like PFK, essentially the idea is. Yeah, essentially the idea with PFK and really any feedback mechanism is you want not Rubisco, you want the cellular respiration. Shh, quiet, please. You want the cellular respiration pathways to be running in such a way where they can receive feedback and adjust things. Meaning, if it needs to go faster, it can go faster. If you want to go slower, it go slower, right? But there are sort of like key points in cellular respiration where it's sort of like a make it or break it moment. And PFK, which is one of the major enzymes in glycolysis, is one of the places where that happens, right? So the idea is that sort of the point usually where they say that cellular respiration is either like a go or it's going to stop. And so, depending on the situation, right, various enzymes or various molecules can provide feedback to PFK. Where, like, if ATP, for example, there's plenty of ATP, that would essentially tell PFK, like, hey, we're okay. You don't have to go so fast. So it sort of might put the brakes on, right? Whereas if there's a lot of ADP, it could be something that says, hey, we need more ATP because there's so much ADP, which means the ATP is being used up. Um, so, in that sense, like it might be then something that is an activator molecule. You don't need to know specifics other than that, like just the diagram in your notes about like feedback mechanisms. It's just sort of the general idea of knowing that there are these sort of key points in cellular respiration where regulation would occur, but you also have multiple points of regulation because it is such a complicated process, which is one of the things I talked about when we talked about the beginning of cellular respiration. The idea that, like, number one, if cellular respiration just broke down glucose in one big step, it would be it would be such a large reaction. It would be essentially be like a explosion inside, right? It'd be too much energy at once. But also having it broken up into those little steps also means you're going to have a situation where it's much more likely to um, have ways of controlling it. But PFK is one of those major control points for cellular respiration. Let's see. Other questions? Let's see. Doing the ethanol. I don't need you to know how they're produced. I would say I want you to know that they are produced, right? I would want you to know that, um, you know, that cells do either or, right? They either do alcoholic fermentation or lactic acid fermentation. That in both cases, it is a way to regenerate your NAD plus since your electron transport chain is shut down. But no, I don't need you to memorize. Like, don't memorize the actual like reactions by which um, ethanol or lactate are produced, right? Basically, if you're able to tell me that you end with pyruvate, and then those pyruvates now, because they have nowhere to go because there's no oxygen, um, then they're going to be converted into ethanol or lactic acid depending on the type of um, cellular respiration or the type of fermentation, right? And to know sort of like who does each type, right? Like we don't do alcoholic fermentation. We do lactic acid fermentation, whereas like bacteria and yeast tend to do. No, thank you, baby. Um, they will do alcoholic fermentation, right? Okay, let's see. Can I go over the citric acid cycle? Yes, I can. Again, remember, I don't need you to know the specifics of the citric acid cycle. What I basically want you to know for the citric acid cycle is the big ideas of it, right? Which would be that the citric acid cycle is the third step. It's sort of like what I equate to like the casino step, meaning that you're going to have all these what you're going to make all these NADHs and FADH2s, right? Because essentially, after the second step, which is pyruvate oxidation, you are going to put that acetyl CoA that you made in the pyruvate oxidation and put it into the citric acid cycle. It's a bunch of enzymes that essentially they continue to break down no, baby, careful. When they break down the glucose further, right, they're going to transfer more energy to essentially NADH and FADH2. And so when you do that, all those NADHs and FADH2s eventually are going to be able to be dropped off to the electron transport chain and be used to make ATP. So you're not making a lot of ATP in the citric acid cycle, but you are generating a lot of the precursors, I guess you could say, or the stuff that can be cashed in for ATP. Yes, okay, cyclic and non-cyclic electron flow. Remember, that is in the light reactions. Careful, baby. That is in the light reactions. In the light reactions, then, with non-cyclic flow, that's what I just talked about, right? The idea that the light shines down, you on photosystem 2 and photosystem 1, they get excited, they get passed down the chain. Oh, I should mention it also for the non-cyclic flow that, once you have that happen, right, to reset everything, then since the electron from photosystem 2 landed in photosystem 1, then you replace the electron in photosystem 2 with water, because you split water through photolysis or photolysis, and then that's how you end up with new electrons to resupply the electron to photosystem 2. So that's non-cyclic flow, right? So in non-cyclic flow, you're producing ATP and NADPH. The difference now for cyclic flow, and remember, plants can kind of switch between cyclic and non-cyclic. Oh, we don't. Okay, it's okay, Mommy, help you. With cyclic flow, what you have there is you have shouldn't say plenty. You have enough NADPH because it's not used up quite as quickly as ATP. So in this case now, the plant cell switches to a situation where it's just basically going to shuttle through the first. Oh, no cake right now. It's going to shuttle through the first. Yes, you have your pretend cake over there. It's going to shuttle through the first electron transport chain. And in doing that, you end up with only making ATP. So in cyclic electron flow, which is usually probably not, all the time, the idea is that when the...
I'm 5.
No, you're not 5, you're 2.
I'm 2.
Yes.
I'm 2.
Okay, thank you. Then you end up with only ATP produced instead of ATP and NADPH.
Yes,
let's see, the gradient in the IMS. I'm assuming you mean in the intermembrane space. Yes, that is going to be a chemical and electrical gradient.
I like that. Can fall.
No, you're fine, Mom. I got you. So yes, it's both an electrical and chemical gradient.
Mommy,
because as the hydrogen...
Your computer's gonna fall.
No, my computer's okay, thank you. As the hydrogens are pumped over onto one side of the intermembrane, then it is a chemical gradient because there's a lot more hydrogens on one side, right? So it's acidic as opposed to basic on the other side. And then it's an electrical gradient because hydrogens have a positive charge. So both of those things will create essentially like a diffusion force that makes the hydrogen want to move back over. Thank you. Well, the chance of you sleeping is slim to none. That's it. Okay, so do both cyclic and non-cyclic flow. I think the two things are separate. I think essentially that a plant adjusts and changes how it's doing things. So if it needs both NADPH and ATP, then it will do non-cyclic flow. And then when it has enough NADPH but still needs more ATP, it will switch to cyclic flow. So I would assume that, by and large, it's probably one or the other at any given moment, depending on the plant's situation.
You can't see me.
You should go to sleep. Let's see. Yes. Okay. Let's see. We talked about photosynthesis, fermentation. Oh, excuse me. Careful. What other questions do you guys have? Oops. Say hi.
Hi.
Yes. Well, on Friday we did a pre-lab quiz, and then we did really quick notes, like 4 slides for like plant land versus water plants, and then they had some problems at work time. So for those of you who, it's quite a few of you who are absent on Friday, you'll be okay. You won't have too much to make up. Okay, Energizer Bunny, sit down, please. Okay, thank you. Sit down. So no, yes, sorry, Max. C3 and cam plants do have the same structure.
Hi.
Can you say hi, Max?
Hi, Max.
So yes, because essentially cam plants separate the processes by time instead of space,
Hi,
then the anatomy for a C4 plant, or sorry, cam plant, is the same as a C3 plant, whereas the C4 special anatomy plant that have those special bundle sheath cells are different. Okay, Hannah, do we need to know about chlorophyll? I don't need you to, I mean, I would say you need to be able to read that graph, like you understand the fact that not all wavelengths of light are used in photosynthesis. But past that, no, I don't expect you to like know it or memorize or anything like that. You are silly. Let's see what are you doing? Can you sit down, please? Sit down. Thank you. Don't fall off the sofa. Then we'll have big problems, huh? Sit down. So. Thank you. Let's see what else. Can I explain the wavelength graph? Yes. So you can sit on my lap. It's fine. Come here. So with the wavelength graph, the idea is that, when we look at light right now, right, like the light that's up there, even though it looks like it's all one color, it's actually not. It's multiple wavelengths. Can you say hi, Hannah? Hi, Hannah. So what happens then is the idea that when you shine light on something, right, not all wavelengths of light are going to be absorbed and used. Some are reflected. And so the idea is that with chlorophyll, it's only able to absorb certain wavelengths of light, namely the red-orange and the blue-purple, but the greenish wavelengths, it really cannot absorb and therefore reflects, right? So the idea is then it's not going to use those wavelengths of light for photosynthesis because those wavelengths light can't reach the chlorophyll. And so there's a correlation then, right, between how much photosynthesis you have for a given how much photosynthesis you would have for a given wavelength of light. Because like for green light, you can see that the oh my goodness, the light is the line is all flat at the bottom, because essentially it means like there's really no photosynthesis happening because none of that wavelength of light is being absorbed. Whereas for like the red orange and the blue green, depending on the specific molecules, right, they absorb slightly different wavelengths of light, then they are going to actually be able to make use of those wavelengths of light for photosynthesis. What else? Anything else? Oh, careful, baby. Careful.
Hi,
okay. Hello. You say hi, sweetie. Okay, really all I need you to know about pyruvate oxidation, it's a very small step, right? I mean, the main purpose of that step is to get you from step 1 to step 3, which is to say after glycolysis we have pyruvate, but pyruvate can't go into the citric acid cycle. So I need an in-between step to essentially remove a carbon dioxide, attach a CoA enzyme, right? So you go from a 3 carbon molecule to a 2 carbon molecule, you lose CO2, you transfer some of the electrons to NADH, and now that acetyl CoA is something that you can now use in the Krebs cycle. And so that's really what I need you to know about. There really is it's a step that gets you from point A to point B. It's also at that point that you're like, or I shouldn't say point A to point C. Um, it's also the step where you are now transitioning into the mitochondria, right? Because if the um, glycolysis happens in the cytoplasm, but everything else pretty much happens in the mitochondria. Let's see. What else?
Hello?
Yes, many hellos from a tired baby.
Hello, Auntie Mama. Hello, Auntie Mommy.
Okay, let me see. What else can I ask
Hello, Auntie Mama.
you? Okay, don't be silly, please.
Hello, Auntie Mommy.
Who's Auntie Mommy?
You're Auntie Mommy.
No, I'm just Mommy.
Hi, Auntie Mama.
Okay, shh.
I'll see you later, Auntie Mama.
Yes, so photosystems are basically, essentially, it's the chlorophyll, but the chlorophyll molecules that surround the chlorophyll. So ultimately, each photosystem, we didn't really go into this in detail, but in case you wanted to know, each photosystem basically absorbs a slightly different wavelength of light. I think it's like, just slightly different, like.
Here.
Okay, thank you, baby. And so what happens then is when pillow, you want the pillow? You can have the pillow. Very pink pillow of your sister's. When you So when you have the photosystems, the central molecule of the photosystem is chlorophyll, right? And so the light's going to shine down on that chlorophyll, and essentially the other molecules will essentially also absorb the light, but then funnel it, like redirect it to the chlorophyll. So ultimately, the electron in the chlorophyll is the one that gets the energy. It gets excited. It jumps to a higher energy level. So they are, when we talk about the chlorophyll, it's really the photosystems, which is like chlorophyll plus their extra entourage of molecules that are going to help them get the oxygen. I'm sorry, the light. Mm. So in the process of making either the alcohol, you still remember Max? Okay. So the way that NAD plus is regenerated is, remember, normally with cellular aspiration, right, you're going to have the NADH, you're going to drop off the NADH, and in dropping off the NADH, you will remake NAD plus. This is crucial because now that NAD plus can go back to glycolysis. Shh, no, thank you. You're going to have NAD plus regenerated that way, right? But the problem now is if you're not doing all of cellular respiration, if you shut down the citric acid cycle, pyruvate oxidation, and the electron transport chain, you can't do that. So, for example, when you take pyruvate and you make it into lactic acid, right, then in doing so, the reaction is not really to make lactic acid, but rather the main thing there is that the reaction itself in doing that will remake NADH or NAD plus. It will take a hydrogen and electrons off of NAD plus H and remake it NAD plus, so that now you have the reactants you need to continue glycolysis. Because fermentation in and of itself doesn't generate any energy. It's really the idea that fermentation is going to allow you to remake your NAD plus, which now allows you to continue glycolysis. That's really kind of the big thing about it. And so all you need to know is that in the process of making the pyruvate, into either alcohol or lactic acid, or in the case of alcoholic fermentation, also carbon dioxide, right? The idea is that in the process of doing those reactions, oh, don't do that, thank you. Okay, hold on. Mommy got it in a second. Okay. Then you would then you end up with the NAD plus instead. You want me to cut it? Oh, okay. I don't know where the nail clippers are. Can you wait 1 second?
It's in my room.
Yeah, we can't get it done because it's too late. Mama find one for you, okay? Hold on. Let's see other questions you guys have. Oh, I guess something to mention then on the subject, as we talk about fermentation, is the idea of aerobic and anaerobic respiration, right? So remember, the whole idea is that you need oxygen as a final electron acceptor in the ETC. And so in that case, when you have that, if you don't have oxygen, essentially the whole ETC shuts down. And when ETC shuts down, then it's going to now go backwards and shut down the Krebs cycle and pyruvate oxidation. So you're only going to do glycolysis. Let's see. How are coupled reactions related to cellular respiration? Essentially, cellular respiration often includes coupled reactions. I mean, it's the overall idea of like when you're making ATP, you're also going to want to have and reactions that use ATP, right? So that's really in the end how coupled reactions relate to that. In that, you see that sometimes in if we were to really break down cellular respiration, but it's also just the bigger idea of energy metabolism. The role of enzymes in glycolysis. So remember, the whole idea behind glycolysis is you're taking glucose and you're breaking it down into two pyruvates. When you do that, that takes enzymes, right? So glycolysis is this 10-step process, and that 10-step process is essentially like, it's almost like molecular origami, right? You're like taking the glucose.
Gummy.
No, thank you. Can you sit down? You're taking that glucose and you're slowly converting it to pyruvate. So every little step is carried out by one of the 10 enzymes in the glycolysis pathway. Okay, what I meant when I said you need oxygen as a final electron acceptor is, remember, when you drop off the electrons, the electron transport chain, right, they get passed down the chain. But at the end of the chain, if you want the chain to keep going and passing electrons, that last protein in the electron transport chain has to be able to give the electron to something, right? The energy from the electrons is being used to pump hydrogen across the membrane. But either way, when you get to the end of the chain, you have to be able to give that electron to something. Otherwise, it would be like a traffic jam, right? So I think we did like a demo in class, right, where it's like if I was to pass something down, but then if the last person in line can't get rid of their electron, then it gets stuck. So oxygen, when you have the electron transport chain, oxygen is your final electron acceptor. Okay, go ahead. You go show them your tea party. So then you end up with you have to give the oxygen to something, or sorry, you have to give the electron to something. So in this case, the electron is given to oxygen along with some hydrogen, and then you end up now with water. So when the electrons are given or accepted by oxygen as our final electron acceptor, then that's where you create water, and that's where the water comes from in terms of the final products of cellular respiration. So the problem now is if you don't have the oxygen, right, everything backs up. It's like a traffic jam. And so molecularly, that's why you actually need oxygen. Like when we breathe oxygen, that's why we need oxygen, because without the oxygen, none of your cells would be able to do cellular respiration because they would have no way of getting rid of their electrons at the end of the electron transport chain. You okay? Okay. 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 Let's see. How often do plants actually go through the I think it really depends, right? I mean, it realistically in most scenarios, they're probably not going through the oxygenase reaction a lot, but it still can happen because the idea is if it's too hot or there's too much oxygen, right? then essentially Rubisco becomes less choosy. Well, I shouldn't say less choosy. It's just not going to be able to react with CO2 as much. And so it will now close up the um, stomata, right? It starts to do the oxygenase reaction. And so I think the figure I gave you guys in class, which is probably pretty close to accurate, is that it can reduce photosynthetic efficiency up to like 25%. So it's probably happening enough that it can be really a damper on photosynthesis. So again, the idea behind like C4 photosynthesis is that these plants have evolved a way to compensate for the fact that otherwise they would probably do the oxygenase reaction more often than they ideally would want to.
Wait, do you have algae?
No, I know. You can put it there. Thank you. All right, let's see. Well, this doesn't fit me. Fits you.
Ow.
Let's see.
Wait, Mommy, you're right. Stay off it. Where's your Katie?
I'm Katie. Oh, okay.
You have to stay
What are
off.
some other types of molecules that can be used in cells? Shouldn't be done. Well, okay, so that's the other thing. Even though we always talk about glucose, it's okay. Even though we always talk about glucose like it's the main molecule, in theory, nearly any biomolecule can be used. And there's a diagram in your notes where we talk about versatility and control, right? But the idea is that because also cellularization is such a multi-step process, then a lot of different molecules can join the process at any given point. So any carbohydrate can pretty much like join into somewhere in the glycolysis pathway, right? Lipids and proteins can be converted into aspects of like I think it's the citric acid cycle and the acetyl-CoA, right? So really, any then biomolecule is probably going to be able to find some way to get into the process and generate some energy. It's just that if we're doing from like start to finish, then we would think of it as mostly glucose, but it's not the case. So why is it the bundle sheath cells? I think based on their so I think it's part of the special anatomy, right? That essentially this cell becomes more of like a closed off to the CO2, and in doing so that means you can like not just the CO2, probably just not very permeable to gases in general. So the idea is that now you're going to be able to create a situation where the CO2 can't just get in and out of the cell, right? Can't get in, but also can't get out. Location in that case is also part of it because it's kind of deeper within the cell or deeper within the leaf. The CO2 is probably not, oxygen is probably not, and CO2 are not getting to that space quite as well as it otherwise would. But in this case, because for C4 special anatomy, if you,
Owie.
it's okay. If you just, find a different way, then you can still create a very sort of high CO2 concentration environment.
Owie.
I know, it's okay. Fix it. It's okay. Let's see. What else?
Yeah, another question. One nail cutter. The nail cutter in my room.
What, the nail clippers? Okay, we'll look for them in a minute. Okay. Let me see if I have any over there.
I'll go check it in my
room. No. Please don't check your room. Go check the table over here. Can you go check the table?
No!
Shh. Okay, sorry guys. Give me 2 seconds. Let me see if I have nail clippers for her. Wait. No, thank you. Mommy has to finish talking to the students here. They're just looking at our empty chair. Okay. Let me check here.
Why are they talking to us?
Because they're studying for a test. Let's see. Any other questions? So it's not that PEP carboxylase itself doesn't smuggle. The idea is that Rubisco being a little less reliable to trap carbon dioxide, since it might also react with oxygen, is going to trap the carbon dioxide as a 4-carbon compound. That 4-carbon compound is not CO2, right? So whereas the bundle sheet cells were not permeable to CO2, they are going to allow the PEP carboxylase to combine to take that molecule, which is called oxaloacetate usually, or malate, depending on the cells, to smuggle in that molecule. So it's not that the PEP carboxylase does the actual smuggling. It's more like the PEP carboxylase traps the CO2 into a 4-carbon compound, and that 4-carbon compound, since it's not CO2, can be, quote, smuggled into the bundle C cells. And once it's in there, it can do a reaction to essentially, release the CO2 back out and use it in the Calvin cycle. So yeah, so PEP carboxylase is something that is, like I said, sort of like it's like something to boost the ability to trap carbon dioxide, since Rubisco does it, but doesn't do it quite as reliably. Let's see, what else can we talk about? Make sure you do look at the practice quizzes. Answer keys to problem sets are in school group. You should be able to find those there as well. We don't have to know. No, you do not need to know the exact numbers of things produced from each cycle. Like I said, I want you to know the idea of what happens. I want you to know what things are made in each cycle, but I don't need you to know exact numbers. How are ATP and NADPH used in the Calvin cycle? So essentially, with the Calvin cycle, what you're doing is you're taking carbon dioxide and you are attaching it to a molecule called RUBP, right? And then you are taking it through a series of conversions. And so, two things to keep in mind, right? Number one, when you have a cycle, one of the things you have to do is... Okay, go get your crayons in.
Mama, this is not all.
Crayons. So with the NADPH and the ATP, essentially what you have to do then is you have to use it in the sense that, like, those reactions that are going to first, remember there's 3 parts of the Calvin cycle. First, it's trap the CO2, right? Second, it's convert that CO2 into a molecule that can make glucose, which is a G3P. And then lastly, after you make some of the G3P, some of the rest of it's going to actually go back to remaking RUBP, so you're ready to receive the next CO2. So those reactions that do all those things use ATP and use the NADPH. And so that's why we say we're going to use the ATP and NADPH produced in light reactions to power the Calvin cycle. Bless you.
I'm going to make a table.
Okay, thank you. Oh, careful. Here, Mommy, help you. On the last slide of the cellular respiration presentation, talked about how cellular respiration is regular feedback. So again, that's just the idea that with cellular respiration, right, it's so important, namely because you're producing ATP, that you want to be able to control it. It's right here. Okay. So you want to be able to control the process, meaning you want to be able to say, hey, I have too much ATP, cellular respiration, you can slow down. And that would be feedback, right? That would be negative feedback, saying essentially, hey, slow down, stop. Or you could have positive feedback, which would be essentially like, hey, there's a lot of ADP or AMP, which means I need more ATP, so you better hurry up and do more cellular respiration. Do you want pink? I can get you pink. Oh, that one's broken, huh?
You have to, there's no pink in it.
Can we find a different color, maybe?
I can use my glitter one.
Okay. So hopefully that makes sense in terms of like feedback. There's tons of like specific.
My glitter one.
Okay.
Working.
Okay, go ahead. There's tons of like processes or specific reactions that are feedback mechanisms in cellular respiration, but you don't need to know those. Just know the general idea. Okay, good.
Little parts.
Let's see. I'm trying to think what else we haven't talked about. Seems like we talked about a lot of stuff.
Golden. Fancy.
Okay, thank you. Let's see. What should you know about leaf anatomy? Really, not a ton. I mean, I would say in this case, just know the idea of like palisade mesophyll cells is where the chloroplasts happen, or where the chloroplasts or where most photosynthesis happens. Know the idea that the C3 versus the C4 plants will have different anatomy, meaning that in the C3 plant, everything happens in the mesophyll cells, whereas in the C4 plants or C4 special anatomy plants, you're going to have the light reactions happening in the mesophyll cells, but then more importantly, the Calvin cycle dark reactions are the ones that are happening in the bundle C cells, whereas a C3 plant, the bundle C cell is there just for structure. Let's see. Other details.
I need other crayons.
Can you go get them yourself?
Okay.
Thank you. Let's see, locations. Yes. So now, assuming we're talking about a eukaryotic cell, right? Because in a prokaryote, it'd be a little different. So I'll talk about that in a minute. In eukaryotic cells, glycolysis happens in the cytoplasm, and then the pyruvate oxidation is where it now starts. You move into the mitochondria. And then from the then the Krebs cycle is going to happen in the very center of the mitochondria, which is the matrix. And then the final step, the oxidative phosphorylation or electron transport chain, is going to happen across the membrane, so also in the mitochondria. If you were talking about a prokaryotic cell, then essentially glycolysis would still happen. I don't know, baby. Glycolysis would still happen in the cytoplasm, but now all the other steps would have to happen either in the cytoplasm or also anytime there's a membrane like the electron transport chain, probably would happen actually in the membrane of the cell itself, of the bacteria, the prokaryote, since it doesn't have a mitochondria. For photosynthesis, as long as we're on the subject, then you would have the light reactions happening in the thylakoid discs, right, of the cells, and then you would have the dark cycle, the Calvin cycle, happening in the stroma, not to be confused with the stomata of the plant. Excuse me, sorry.
It's stuck.
What's stuck?
My fingers.
Oh, your finger. Oh, why is your finger there?
Ow.
Oh, don't go for that. Here. This way. So, take the ones you want, huh? Sorry. Sorry, it's very eventful today here. Anything else you guys wondering about? Let me think if there's anything I would want you guys to know that I haven't thought about yet. I think I've already talked about anaerobic and aerobic.
What is it, Mommy?
Well, Mommy's just talking to students.
Your students? I probably get them.
Okay.
Let's see. I'll just get a chair.
Let's see. We've got the different types of fermentation. C3 versus C4 photosynthesis. Remember that in photosynthesis, the electron transport chain is essentially the same idea of as in cellular. So maybe really quickly to review the ideas that you drop off the electrons.
Talking about your students.
Mommy's talking to my students, not about them. So with the electron transport chain, remember that is you drop off the electron. Thank you. You drop off the electrons, right? The energy from the electrons is going to be used to create the gradient, which it pumps. That's okay. Pumps the hydrogens across the membrane, right? Now that you have this imbalance of hydrogen, now hydrogen wants to come back across to balance it out. The only way it can do that is it goes through the doorway. The only doorway it can go through is the ATP synthase. So as it goes through the ATP synthase, it actually rotates the molecule in such a way to produce ATP. Okay, good. Let's see. Hi, Arush. Okay, so let's see. Anything else?
Yeah, let's see.
Maybe, another quick thing to just keep in mind
Let's see.
is that, remember, we talk about cell respiration and photosynthesis.
Let's see, Mama.
Okay, good job, baby. But remember, plants do both, so don't sort of neglect, I guess, for lack of a better word, that idea that plants are doing both cell respiration and photosynthesis, whereas we are only doing cell respiration. Because some people forget the whole plants and cell respiration part. That's okay. By accident, it's okay. Okay, so any other questions? Looks like we have a couple minutes left, but I don't know if we've exhausted the questions.
Let
me see. Not sure if I'll be able to successfully pull any PowerPoints up on my computer, but we'll see. Maybe I'll be able to look at it for a second and see if there's anything here you guys might need. It's opening all sorts of things. Okay.
paper. I want a big paper.
You want a big paper? Okay, do you have a big paper? Oh, no, and again, Arush, you don't need to know the specifics of how many are produced. Just know the big ideas. I want you to know what goes in, what comes out.
Showing a big paper.
Okay, mommy can try to. Here, can you have a little paper? How about this paper? Okay. Okay.
I'm gonna glue on it.
Okay.
This.
Oh, you just colored your chair. Thank you.
Huh? What is it?
Don't color the chair, please. Just color the paper. All right. Any remaining questions? Let me see if I pull up the PowerPoint, if anything seems to. Hmm.
Here you go.
Thank you, baby.
I want another paper.
May I please have another paper?
To glue it on.
Please. Let's see.
I still want to.
How effective are other energy sources like fats? I mean, I think they're still effective. I think the idea is that, you know, you have to, they probably don't have quite the same, in the end, if you're making ATP, it's something, right? But I think the idea is that carbohydrates tend to be the source of like immediate energy because they're really easy to like break down, whereas like the fats, you have to break them down into something else, then put it into the cellular respiration pathway. So could it be done? Of course, happens all the time, but it's a bit more roundabout.
Another.
Please. The RUBP, remember, is essentially the substrate of the Rubisco. So
this is no name.
That's okay. So you're basically going to take that RUBP, attach a carbon dioxide to it. That RUBP is a 5-carbon molecule. So now that you attach a carbon dioxide, you're going to split that 6-carbon molecule into 2 3-carbon molecules, and those will go through the rest of the Calvin cycle. Again, remember with cycles, part of it is to generate something, right? Like the molecule becomes glucose. But then you also have to like regenerate the RUBP or whatever starting molecule you have so that you can have something to meet the next CO2 as it comes in.
Give me the other one. Give me that.
Please. Okay, let me check my PowerPoint again, but again, feel free to keep putting giving me questions if you've got them. I'm just trying to see if there's anything we missed. Let's see. Don't worry. I guess something I could tell you not to worry about is in your lecture, we talked about really briefly, like free energy and that equation for like enthalpy and entropy. Like you definitely don't need to know that. So don't worry about that. But you should still know like exergon and antergon, like that kind of thing.
Another one.
Yeah, is it the last one? Okay, baby. Here you go.
I don't have any names.
Yes, I can give an example of cam plants. Pineapple plants are a good example of a cam plant. So they are the ones who are going to, where it's like sugarcane is a good example of a C4 special anatomy plant, right? Different strategy, same idea is find a way to trap more CO2. So the cam plant will do that during the nighttime. And so essentially stomata are closed during the daytime because you've had all this CO2 trapped during the nighttime. You have to worry about triggering photorespiration. Okay, looks like we're coming up on like 1 minute left.
Mommy, this is a broken.
No problem. Sorry, it was a little delayed. I was not expecting my computer to have issues, but restarting it helped. You're welcome, guys. Well, if you guys don't have any other questions, I think what we'll do is maybe cut this off a tiny bit early so I can get her to sleep, because even though she doesn't think she's tired. Baby, come here. You want to say bye-bye to the students?
Bye-bye, students.
Can you say bye-bye?
Bye-bye, students.
Okay.
I said bye-bye.
Yeah, good job. All right, guys. If more questions come up, yes, I hope she falls asleep quickly. I think she will. She's my night owl, apparently. So basically, if you guys have more questions today, tomorrow, come find me, or come find me on Monday, or you can also email me over the weekend, and I'll be checking stuff. So thank you, baby.
Yeah.
All right. So I will see you guys later. You all should go to sleep too now, because big kids need sleep as much as little kids, right? All right. Have a good rest of your weekend, and thank you, baby. Let me know if you guys have any other questions. All right. Okay. Bye-bye.
Bye-bye.
Bye-bye. Say
Bye-bye.
good night.
Good night.
Okay. Bye-bye.
Bye.